So, good morning, everybody. I'm Sonia, and the title of the project that I'm going to present you today is the role of CKM44 in the dorsal lateral geniculate nucleus. So in our lab, we are interested in AMPA receptor auxiliary subunit. There are a particular class of protein that affect the trafficking and the gating of AMPA receptor on the cell membrane. And in particular, we are interested in the protein that is so-called CKM44. And as you can see in the bottom line, is nicely expressed in the dentate gyrus as well as in the DLGN. And these are the results from high in situ hybridization experiments that have been performed in our lab a few years ago. In particular, what has been observed is that the deletion of the protein leads to a faster recovery from desensitization. And you can appreciate here from the recovery tau, so the quantification, and you can observe that this value is significantly reduced when the protein is deleted. Moreover, when stimulating the optic tract, it was observed that the pay-pulse ratio in the knockout animal was significantly increased compared to the wild type, suggesting that the short-term plasticity is strongly influenced by the deletion of the protein. After that, one of the main questions was whether the synaptic input integration was affected also by the deletion of the protein. So performing current camp experiment was observed that not only the amplitude of the EPSP was affected when the protein was not there, but also the firing rate probability was increased in the knockout animals, suggesting in this way that when we stimulate the optic tract with high frequency, we should observe the big effect due to the deletion of the protein. So the main question, that would lead to, so it's the big picture. So the main reason why we want to model this network is because we want to understand what is really the interplay between the receptor desensitization and the glutamate spillover that occurs in the synaptic cleft during cellular signaling, because this is also another kind of mechanism that it's really relevant. But for the purpose of the project that I have been working on this week, I will just focus on the AMPA receptor desensitization role. So let's just jump into the network that I developed. So, okay, so let's start from the top. So we have our external input that I modeled as NetStim, and the input will be projected directly on a populations of constituted by retinal ganglion cell that here are represented just by a simple kind of ballistic model with very long axon that then will project directly on the population constituted by 20 relay cells, and this will be realistic morphology. Simultaneously, the relay cells receives input from a population of 5 interneurons that are modeled in the same way I did with the retinal ganglion cells. So the connection. So the red lines here represent the excitatory inputs, and the blue line represent the inhibitory inputs. And so the connection between retinal ganglion cell and interneurons relay cells, I model as just a simple mode file, so a double exponential. It's the well-known, so we worked with that during the weeks, X2 whereas the connection between retinal ganglion cell and relay cell, I modeled in a different way. I used a release mode file that was developed by Destexe and colleagues in the early 90s. And this basically is a mode file where a cascade of events that involve calcium influx in the cell leads to the production, let's say, of glutamate. The glutamate then will point to our customized AMPA receptor model. I will tell you in a second about that later. And of course, then this will lead to the activation of relay cells. So let's talk about the AMPA receptor models. This is a model I have been working on since, I don't know, maybe 4 or 5 months. And so I implemented this 9-state kinetics. And yeah, with the aim to reproduce our experimental result from fast application experiments, where we stimulate the nucleated patch with a 1 millisecond glutamate pulse. And as you can see, by using a function that is implemented in neuron, it's called the multi-run fitting, sorry, I don't remember exactly at this moment the precise name. I have been able to reproduce the experimental data, nicely fitting the experimental result from the peak and the decay time. Also, you can appreciate here how the results from the model can reproduce our experimental data. So these are the experiments, so the recovery from the sensitization observed in the wild type, and this will be the computational model related to that, and this is for our knockout animals. And in the quantification, you can see that the recovery tau from the model that is indicated in the dashed line nicely fit the experimental data. So once our AMPA receptor model is ready, we can put that in our morphological realistic reconstruction of relay cell, and we can start our experiments. So I decided to perform experiment at 20 Hertz frequency stimulation. So what that leads to, so we have the activation of our retinal ganglion cell. Since our retinal ganglion cells are connected to the interneurons, we have just 1 or 2 milliseconds afterwards, we have activation of interneurons. Simultaneously, since I said that in the retinal ganglion cell I implemented the release mode file, so the mechanism that leads to the production of glutamate, we have here the release, let's say, of glutamate that then will point to our relay cell model. And then the combination of these two inputs leads to the response of our neurons. And this is the excitatory postsynaptic potential curve that it's obtained. So this is, let's say, valid for our wild type cells. So, but if we want to see what the effect due to the deletion of the protein, we need to switch also to our knockout model of the AMPA receptor. And as you can see, the excitatory postsynaptic potential increases over time and upon stimulation. And this is, of course, is not quantitatively in agreement, let's say, with the experimental traces, but still you can see that the trend, let's say, is reproduced. And this is okay, this is good for the excitatory postsynaptic potential amplitude, but also when we look at the Fari rate probability, we can see that if in the wild type animal we see an action potential just at the beginning of our EPSP curve by increasing a little bit the stimulus strength, when we switch to our knockout, we can see that the Fari rate probability, the Fari rate is maintained, let's say, throughout the entire time of recording. And again, this is in agreement with the trend that was experimentally observed. Last but not least, thanks to the way our AMPA receptor model was implemented, we had access also to the portion of the AMPA receptor that are in the open state during the stimulation. So in here, you observe that the 40% of AMPA receptor are activated at the first stimulus, and about 12% will be activated on the second stimulus. But if we look at the knockout animals, we can see that already, first of all, at the first stimulus, we have an increase in the portion of receptors that gets activated, but the second stimulus, the number of AMPA receptor activated is much higher compared to the wild type. And this is, again, in a qualitatively, let's say, agreement with the experimental results that we obtain from our pair-pass ratio, since we see that the pair-pass ratio in this case, is 30 milliseconds, but if we should be in some place here. So it's significantly increased compared to our wild type animals. So in the end, we saw that our wild type and CKM44 AMPA receptor models are quite good because they can reproduce a nicely fit experimental result from the ultrafast application experiments. So we also observed that if we just implemented in our network this customized receptor model, we can partially reproduce the difference that was observed both in the synaptic input integration and in the firing rate probability. And this is valid also for the peak open probability. But of course, these are preliminary results, and this show that the faster desensitization cannot alone, of course, fully explain the effect due to the deletion of the protein, suggesting that another mechanism has to be implemented in the network, and suggesting that this mechanism would be glutamate spillover and this is known from the literature, suggesting that, of course, to fully evaluate the role that is played by our CKM44 protein, we really need to implement the spillover mechanism. So the diffusion of glutamate in the neighboring synapse has to be absolutely implemented in the network. So last but not least, I want to thank all the people involved in this course. Has been really, I had really nice time, really interesting talks. I learned a lot. And of course, I want to thank Craig because he was really helpful to me during this week. He helped me a lot. In the end, thank you all for your attention.
Great job.
Yes, great job, Sonia. Thank you very much. Okay, any questions for Sonia here?
And how do you think, sorry, let me open my camera. So what do you think you have to tweak in the model so you can like not only have the qualitatively similar results, but now actually try to like match the quantities in terms of the EPSP amplitude? Do you have any idea on like which parameters you should start with?
Yeah, I should start working with the diffusion exactly because as being observed, I mean, there were other models that have been developed over time, but they use a different simulation environment, and they showed actually that of course, working just, for example, with two synapses, they showed that if we have just a small spillover of glutamate, you can explain somehow the different shape that you can observe in the APSP amplitude. So yeah, of course, probably similar results could be obtained also by using simple, for example, X2CN or tuning a little bit that mode. But of course, this is completely different for the purpose of, let's say, the project. So I really need to work with the diffusion, and you might play around with, I don't know, the diffusion constant, the tortuosity, the fractional volume, and also the distance, for example, and the synaptic cleft. Yeah, there are a lot of parameters. Some of them are available, of course, in the literature, so one should start with those, trying to tune a little bit what you know can be tuned, and then you see.
Yeah, we often have a shortage of the data we would love to have, and so you find the best data set available, and you work with that, and you try to encourage experimentalists to collect what you're interested in.
Yeah, exactly.
Well, thank you very much, Sonia. That was fantastic.
Thank you.
And coming